Hello everyone, welcome to another new video. Today we will discuss about the bacterial growth curve. In the previous classes, we have understood that what are the morphology of bacteria. We have understood morphology, how they all look like, and after that we have understood the introduction of the bacteriology properly. And today is the third class of microbiology. We are going to understand the growth curve, G for growth curve. Okay, it is important to understand how the bacteria become grow. As I have skipped one class, that is growth and nutrition of bacteria. I will say you that which kind of factor or which kind of things are required for a bacterial growth. They require some kinds of carbon, oxygen, sulfur, some kinds of minerals or phosphorus. These kinds of whole things are required for an bacterial growth. Okay, and whenever they are getting these kinds of ingredients where they are needed, then the growth curve becomes started. So that if you are noticing here, one phase is called lag phase. So that if I am writing here, one is lag phase and another one is the log phase, another one is the stationary phase, another one is the phase of decline or death. Okay, there is basically four phases of bacteria growth, bacterial growth. Just notice here into the lag phase here, bacteria, we are understanding after today's class, you will not face any kinds of doubt and you will never forget that how the bacteria it become grow. See, first is the lag phase. Into the lag phase, see here, if you are noticing here, into the lag phase here, uh, does not any kinds of growth are occurring. Into the lag phase, their bacteria are accumulating their effort, accumulating their whole kinds of nutrients for the bacterial growth. Second is the preparation for growth. That is the lag phase. Here, bacteria are accumulating their ingredients and taking the preparation for the growth of bacteria, for the growth. Okay, these kinds of two things are occurring into the lag phase. After that, if I am coming to the log phase here, bacteria are already getting the whole things or whole kinds of ingredient into the lag phase. So that into the log phase, here is a growth. Growth are occurring exponentially means they are giving their 200% for growing. See here does not require any kinds of bacterial growth, but here is a bacteria become grow. See here, if you are noticing the curve, here is the bacteria become grow. These kinds of growth are occurring into the log phase. Here the bacterial growth are occurring exponentially. Maximum number of growth is occurring within the log phase. Understood? After that, see here, that is a stationary phase. When you are noticing the station into the train station, that is not moving, right? Train is moving, but station is not moving. Here is also into the stationary phase, bacterial growth become stop, but they are not decreasing. Here, if you are noticing here bacteria are accumulating which kind of ingredients is required into the lag phase. Into the log phase, they are utilizing this kind of nutrients and grow their cell into the log phase so that growth is occurred. And into the stationary phase, they are not growing and they are not decreasing. They are just sustained in their position. That is called stationary phase. It does not require any kinds of growth. So that into the growth are not happening, decreases are not happening into the stationary phase. Okay, here our bacteria are not able to grow in their cell. After that, there was also phase of decline. That is called phase of decline. Decline means deny. Decline means stop. Decline means the giving the negative response. That is phase of decline. Into the phase of decline, here whole kinds of nutrients or ingredients become finished, so that the bacteria have to die. So that bacteria are not able to grow, right? So that bacterial whole kinds of ingredients become completed as well as become finished, so the bacteria become decreases. That is called phase of decline. Before the death, there was a phase of decline. Phase of decline here does not require any kinds of ingredients. Here does not require any kinds of nutrition. Here is the bacteria become decreases due to lack of nutrition, lack of oxygen, lack of ingredients. Okay, and after that last is the every cycle have their growth. We are human being, we have to also death, right? We have to also die. We was born and we have to die. Okay, here is a bacteria is also death occurring. Bacteria become death. Bacteria become death. Okay, that's all are the bacterial parts. If you are noticing here, here is a, if I am sum upping the whole things, there is a lag phase, log phase, stationary phase, phase of decline, and the phase of death. Death means here bacteria become decreases. So that the question, first question is in which phase bacteria become grow too much, then your answer should be log phase. In which phase bacteria are not growing and not decreasing, your answer should be stationary phase. In which phase bacteria become death, then your answer should be after phase of decline or into the death phase. Okay, and there is a time, that is a time become increases and bacterial growth become sometime increases, after that they become decreases. That is a number of bacteria. Okay, and whenever we are using the culture media for growing the bacteria, that is also these kinds of phenomena are occurring. Okay, and whenever we are seeing the bacteria, we are seeing the bacteria, if they are viable or viable, defining that they are visible or they are alive in bacteria, that is called viable bacteria. Okay, that is the live bacteria which are living. Okay, that is a viable bacteria. Understood? So that's all about the growth curve. If you are facing any kinds of doubt, just write down into the comment box. I will give your answer. Just stay tuned with us for new classes. Please subscribe our channel and join our social platforms that will enhance your lab technology journey. Thanks a lot.